invitation. As Rita mentioned, my lab has had a long-standing interest in understanding how the brain regulates bodily functions. Particularly, we've been interested in metabolism because metabolism and metabolic dysregulation sit as a node of many diseases that are adult onset diseases or diseases of aging, including diabetes, cardiovascular disease and strokes, certain cancers, even neurodegeneration. And so our thinking is that if we can understand how metabolism is regulated and what happens when it goes awry, then we may be able to get a hook into the sort of very core of what happens, what goes awry as we age. More recently, as I'll show some very early data on, we started off with this sort of the brain regulates bodily functions, but our data over and over again started pointing to the converse as well, which is the gut, one of the most important organs in multicellular organisms, and certainly for C. elegans, is also sending signals back to the brain. And so we've become very fascinated in what these sort of different peripheral organs, what information they may be relaying to the brain. And so before I jump into the data, I would like to give a little bit of a sort of theoretical underpinnings for sort of how we think and how we come to study these problems and the way that we study them. And so for that, I want to start by first noting that one of the striking features of humans around the world is the incredible diversity that we see in aspects of physiology. For example, height, body weight, body structure, that sort of thing. And when it comes to metabolism, we think that in the 21st century, we are in this very kind of obesogenic environment, and that propels us towards obesity and metabolic dysregulation, the accumulation of body fat. But in fact, obesity and leanness have always coexisted. And this is perhaps best visualized by looking at art through the ages, whether it's going all the way back to the Venus of Willendorf that is dated back to 20,000 BC, or art from the 1300s down to the sort of roughly 1800s, where obesity has always been depicted in coexistence with leanness. In other words, in modern parlance, in the post-Mendelian and McClintock eras, we would say that obesity is a heritable trait. So these general sort of artistic observations began to be codified in the scientific literature in the late 1800s and the early 1900s, in which the idea of homeostasis was evoked, right? So this was pioneered by Claude Bernard, Walter Cannon, and others who they were very influenced by engineering control theory, and they invoked what they called a set point model for many homeostatic mechanisms, which was originally posited for temperature and blood glucose and so on, but then later was included to incorporate body fat. And the idea essentially is this, which is body fat is maintained at a certain level. If there is an intervention in some way, so for example, a high calorie diet, the set point model would say that there is a slight increase in response to this external change from the environment, but that an internal homeostasis mechanism would take hold and it would stabilize. When the animal is taken off the high calorie diet, again, there is a dip to make an adjustment for the environment, but then the homeostatic system returns. Now, work from the 1950s onwards by Kennedy, Wirtschafter, Davis, and others, using rodent models, pretty soon put this notion of the set point model to rest, right? Because experimental observations simply didn't model that. And so that in turn gave rise to what they call the settling point model, in which they said, body fat, again, on a high calorie diet, an animal will continue to increase its body fat stores until it is removed, at which point if the animal's body fat sort of goes into free fall. And again, experimental observations did not match the settling point model. And not unsurprisingly, perhaps, people came up with this sort of hybrid model where they said on a high calorie diet, body fat can increase up until some threshold value. And when the animal is taken off that diet, then there is a lot of variation right? This is kind of where we are now. There's profound variation in the degree and extent of body fat loss when the animal is taken off the high-calorie diet. Now, both from spontaneous mutations as well as the pioneering work for the discovery of insulin as well as leptin, we now know that there are anabolic hormones that are responsible for these effects. But catabolic hormones that bring an animal back towards homeostasis have not really been discovered. And this is not just an academic debate about what's the right model for body weight homeostasis, set points, settling points notwithstanding. It's a very practical and urgent matter because worldwide, the genetics notwithstanding, obesity has tripled since the 1970s. And metabolic derangements, as I mentioned, are at the root of a host of illnesses. And so if we can understand the particular combinations of genetics and the environment that lead to this variation, we may be able to make some progress, both from a management standpoint as well as from a therapeutic standpoint. And so when I first started thinking about this problem, I was very interested in coming up with some way in which we could analyze multiple genes at the same time, look at properties of entire networks, but without losing mechanistic resolution. And for a variety of reasons, settled on the C. elegans model system. And for those of you that may not be so familiar with it, I'll just quickly introduce you to the system. It's a tiny microscopic worm. Its cell lineage is invariant. We know exactly how many cells it's made up of. It's it's made up of about 1,000 cells, 300 of which are neurons. One of the great advantages of the animal is that the body is transparent, and the animal is made up of 10 pairs of intestinal cells. This is towards the nose of the animal. This is the pharynx, the feeding organ, and we'll get more into the neurons and the nervous system, but that's not visible here. Body fat is stored in a single organ, in the intestine, which is 10 pairs of cells, and you can see the intestinal lipid droplets visualized here. Fat metabolism is very quick and labile, right? So an animal that is fed and then taken off food within 3 hours will deplete close to 80+% of its body fat stores. And these physiologic changes can be visualized at the molecular level by using reporters, which we've developed over the years, in which we can see, for example, one of the canonical rate-limiting lipases called adipocyte triglyceride lipase that is responsible for converting stored fats into individual free fatty acids that then get metabolized for energy. That enzyme is present largely in the intestine. There's some robust expression in the feeding organ as well, but a fasted animal will upregulate transcriptionally this enzyme in order to cope with this conversion of stored fat for energy. We have a pretty good way to visualize the accumulation of fat stores as well, because an animal that is actively restoring its fat stores will upregulate an enzyme called FAT7. And so what this does is allow us at a very large scale to conduct genetic and genomic screens to be able to identify with no biases ahead of time genes that are involved in regulating fat metabolism. So that's as far as the sort of metabolic side of things, right? Now, what about the neurobiology side? I mentioned that we're very interested in how the brain regulates body metabolism. One of the early things that I had discovered when I was a postdoc in Kaveh Ashrafi's lab was that distinct neural pathways regulate food intake and fat metabolism. And we had found this in the context of serotonin signaling. And I'll talk more about that in a minute. But what that really means is that the neural circuits that regulate food intake and fat metabolism are completely distinct from one another. And that's surprising, given that there's such a strong general perception that metabolism is really just a function of calories in and calories out. But our sort of genetic analysis was indicating that it was not as simple as that. And so it sort of gives us, the neurobiology then gives us a hook into understanding at a more global, systematic, and mechanistic level the ways in which the nervous system regulates metabolism. Other advantages of using the system are that because of the pioneering work of others in the C. elegans neuroscience field, a lot is known about individual promoters into specific neurons of interest that are expressed in specific neurons of interest. Additionally, sensory modalities that are regulated by individual neurons is also known. And so if you combine those two together, if you get a hit in a genetic screen for a gene that's expressed in neurons, you can be somewhat confident that you can immediately, if you can identify which neuron that gene is expressed in and is required in, you can start to take guesses about which sensory modality is involved in regulating metabolism. So it gives you a very powerful way to start coupling different sensory mechanisms and different ways in which the world, the environment for the worm gets interpreted to regulate metabolism. So that's really the sort of power and the motivation for using this particular system. There's other advantages. The C. elegans gut is not directly innervated, which means that any effect of the nervous system on the intestine has to be conveyed through endocrine means. And this gives us an opportunity to discover new endocrine mechanisms that are involved in the catalysis of body fat stores. As I mentioned, for the hybrid model, right, how fat breakdown is regulated is actually still very poorly understood. And so this gives us an opportunity to sort of fill that gap. So in the work that we've done so far, we've screened approximately 30% of the estimated neuronal genome, perhaps a little bit more by now, around 10 to 15% of the total genome. So I'll tell you a couple of stories about what we found, and then talk sort of about the gut to brain connection. So one of the first screens that we did was for the G alpha family of proteins, which are proteins that sit downstream of GPCRs and are involved in controlling cell function physiology. The G alpha family in C. elegans is elaborated. There's a lot of members. There are 19 members, of which only 1 or 2 are lethal. So we simply took loss of function mutants of the loss of function GPA mutants and asked how many of them have changes in body fat stores. The other advantage of this family is that the exact neuronal anatomy of every one of these genes is known thanks to the work of others. We got 2 hits from the screen. One of them is called GPA8, the other is called GPA3, and I'm going to tell you a little bit more about GPA8. We were immediately fascinated by this because of its anatomy. So it turns out that GPA8 is a G-alpha protein that belongs to the Gus-Dusan family of proteins, right? So it regulates intercellular functions by controlling the second messenger cyclic GMP. Now, GPA8 is only expressed in very few neurons. It's expressed in those neurons that have 2 very interesting properties. One, they can only sense and detect the internal milieu of the animal. That's why they're called the body cavity neurons. That itself is fascinating. Second, these neurons were known from the work of Bargman and De Bono and others to be sensors of environmental oxygen. So that was sort of instantly fascinating. We noticed that the changes in body fat in the GPA8 null mutants was not because they had any change in food intake. In fact, what we saw is that these mutants had a constitutive increase in the enzyme in the gut that converts stored fat to usable energy, right? So these animals are constantly metabolizing their stored body fat. And we thought that was fascinating. Now, what is so important about these oxygen sensing neurons? How might this be working, right? So that's the question that we were interested in answering. It turns out that the biology of the DRX neurons is completely fascinating. So if you give C. elegans a range of oxygen from 0% to 21%, right, which is atmospheric oxygen, they can detect and respond to that range and prefer to be in a range of oxygen in the 10 to 15% range that's shown here, right? So wild type animals like to be in the 10 to 15% range of oxygen. And it's because worms eat bacteria and bacteria respire and drop the local ambient concentration of oxygen from 21% down to around the 10 to 15% range. So it turns out that the 10 to 15% range, which is completely distinct from hypoxia, right? This is all in the normoxic range, is an indicator of respiring bacteria, which represents an excellent food source for the animal. And so we thought to ourselves, okay, well, what actually happens to fat metabolism in these two different ranges, right? And so we did some experiments in which we took fed worms and then we fasted them at either 21%, right? So just on the bench top, for 2.5 hours. And as I mentioned, within about 3 hours, worms have lost 80+% of their body fat stores. So fat metabolism is sort of very quick. So we either fast them on the bench at 21% or we fast them at 10%, right? Which is a normoxic range, but this is a range that represents availability of food. And we simply ask the question, what actually happens to their fat stores, right? And so it turns out that if you fast animals, they lose about 80% of their fat at 21%, but they retain 2 to 3-fold more fat at 10%. And so we thought that was fascinating. That change in fat is not because the 21% animals are sick or because if there's any change in food intake. There actually isn't. It happens because at 21%, the animals are able to turn over their fat for energy at a higher rate than they are at 10%. Now, what do the oxygen sensing neurons have to do with this, right? Is this controlled in the gut? Is this controlled in neurons? And it turns out that the oxygen sensors are already known. The predominant oxygen sensor for these particular neurons is called GCY36. It's a soluble guanylyl cyclase, which binds molecular oxygen through a heme group and generates cyclic GMP in proportion to the oxygen that's bound. It turns out that GCY36 mutants, right, that are missing this sensor are not able to burn fat at 21% in the same way as wild types. So their fat burning looks more like what you would see in the 10%, suggesting that the oxygen sensing from these neurons is what is regulating fat stores in the intestine. So what about the mutant that we originally discovered in the screen? It turns out that GPA8 mutants have the opposite phenotype, right? They, first of all, are always burning more fat, right? That was the original phenotype that we found. But whether you put them at 10 or 21%, they're burning fat as if they're in 21% all the time, right? They don't get that the animal is switching, just like the GCY36 animals don't, but the direction is the opposite. Double mutants look like GCY36, and the epistasis essentially suggests that GPA8 is most likely a negative regulator of GCY36. And, without taking you through sort of all of the genetics and the sort of epistasis and how we figured this pathway out, I'll just bring you to the crux of it, which is that it turns out that from doing GCAMP experiments by placing animals in a microfluidic device where we are able to give them either 10 or 21% oxygen, we see that GPA8 mutants have a reduced response. But it turns out that the altered and reduced response of these mutants is because their median baseline of URX activation is much higher. So in other words, these URX neurons are always on in the GPA8 mutants. And so that gave us sort of some explanation for why these GPA8 mutants are always burning fat. URX neurons are always more on. It stimulates this enzyme that converts stored fats to energy in the gut. Now, while we were working this out, another postdoc in the lab, Rosalind, became interested in neurons that control the opposite response, right? So I told you that these URX oxygen sensors respond to quote unquote high oxygen, right? They're responders of 21% oxygen. But another pair of neurons called the bag neurons do the opposite, which is they keep animals away from very low responses. So hypoxia in the worm, the HIF pathway is activated at 3% or lower. But the bag neurons are responsible for keeping worms away from sort of the 5% range, right? So there's a push against 21% by the URX and the push against 5% and below by the bag neurons, so that together the animals stay in that 10 to 15% range. The oxygen sensor that regulates the bag responses is called GCY33. And we sort of became naturally curious in the guanylyl cyclases because of our previous work with the URX neurons. And we found that a particular guanylyl cyclase that's robustly expressed in the bag neurons had reduced body fat stores, and none of the others did. And through necessity and sufficiency experiments, we found that GCY33 in bag neurons was responsible for the reduced body fat phenotype. I won't take you through too much of this, but you can see again, it's a similar set of experiments. We fast the worms, and now we sort of see the opposite, right? So the GCY36 in the URX neurons was responsible for blocking the 21% response, but the GCY33 in the bag neurons is responsible for blocking the low 10% response. The double mutants look like GCY36, giving us some indication of the relationship between these two different neurons in how fat metabolism is ultimately regulated in response to environmental oxygen. If you remove the bag neurons altogether, there's a severe decrease in body fat stores. So of course we had to go in and look at what was happening at the neuron level itself. And so when we express GCAMP in the bag sensory neurons, when we express GCAMP in both the bag sensory neurons as well as in the URX neurons, what we found is that ablation of the bag neurons completely abolishes the URX responses, right? And this is again because there's a profound increase in the URX resting potential to the point where environmental oxygen is now no longer able to evoke a response. So I will not have time to take you through sort of how this communication works and how we found what the mechanisms were, but it turns out that there is a neuropeptide that is released from bag neurons. It's called SWIP17, and it activates under low oxygen conditions. It works to repress URX neurons through its cognate GPCR, and that this relationship between bag and URX in response to fluctuations in environmental oxygen is the major mechanism by which fat in the intestine is regulated. Now, while all of this oxygen work was going on, we had continued our initial work on serotonin, which is of long-standing interest and near and dear to my heart because it's one of those fascinating molecules that seems to regulate everything, not just in worms, but in mammals as well, right? So it's involved in the control of mood, it's involved in the control of behavior, it's involved in the control of metabolism, it's involved in gut functions, it's involved in immune responses. It just seems to be one of those sort of does everything molecules. And the question then becomes, well, how does it affect any one individual output without affecting everything else? Right, so we've been very fascinated with this and had observed a long time ago. So serotonin itself in C. elegans is made in only a few pairs of neurons. And one of the neuron pairs that is exquisitely responsive to changes in food availability is called ADF. The name is not important, but the salient feature of those neurons is that they express TPH1, which is the rate-limiting enzyme for the production of serotonin. Well-fed worms express robustly TPH1. This is a TPH1 reporter. Fasted worms show a profound decrease in expression levels of TPH in the ADF neurons, and then when they're refed, we see TPH expression come right back on. So we had observed that endogenous phenotypes of neuronal serotonin deficiency in excess gave rise to changes in gut phenotypes, again, for metabolism, right? So MOD5 is the name of the gene that regulates serotonin reuptake, right? So the serotonin reuptake transporter is called MOD5. MOD5 mutants recapitulate constitutive excess of synaptic serotonin. And you can see a subtle but clear decrease in body fat stores. So increase in synaptic serotonin, leads to decreased body fat stores. In contrast, TPH mutants that don't make any serotonin at all have a slight increase in body fat stores. So these are the endogenous phenotypes of serotonin. These phenotypes can be very powerfully mimicked and recapitulated by exogenous administration of serotonin, which you can see here dramatically decreases and reliably decreases body fat stores. And this doesn't happen because the animal is sick or there's any type of malaise. In fact, we've looked and we can't even see a clear change in lifespan in animals that have been treated for serotonin. This decrease in body fat store turns out to actually result as a consequence of increased oxygen consumption, which is a surrogate for energy expenditure. So these animals are losing their body fat stores because they're simply expending more energy, right? So using this exogenous administration of serotonin then becomes a very powerful handle for us to be able to figure out the genetics of how neuronal serotonin and presence of absence of food regulates body fat stores, right? And so through a series of reconstitution studies as well as genetic screens, we worked out the neural circuit for serotonin-mediated fat mobilization. And this is older published work, so I won't get into it too much, but we essentially discovered that there is a synergistic relationship between serotonin and the invertebrate ortholog of noradrenaline called octopamine, and that these two neurotransmitters function together and in combination actually with the URX neurons to regulate fat mobilization via ATGL activation. In work that I won't have time to show you, we also discovered a role for population density sensing via the pheromone called ascaroside-3 or ASCAR3. And it turns out that ASCAR3 is detected by pheromone sensing neurons called ADL, in which the other G alpha protein that had come out of our screen, GPA3, is both necessary and sufficient and functions via acetylcholine, again, to regulate ATGL. What does this really mean, right? What we think is happening is that the worm is an exquisite sensor of its environment, right? So it's using a combination of oxygen, both as an indicator of food availability, and of course, oxygen is essential for all metabolic processes, particularly fat metabolism. And so it's using the sensing of environmental oxygen by using oxygen sensing neurons, but that information is integrated with a different indicator of food availability, serotonin. But what's pheromone sensing? What does pheromone sensing have to do with it? And it turns out that this particular pheromone, ASCAR3, is secreted only by adults, and it's an indication of population density. So C. elegans is a lot more sophisticated than I might have ever imagined. Not only can they tell how much oxygen and food is present, they are gauging how many of one another there are using the pheromone ASCAR3 to make a calculation for the rate and extent of fat loss. So in settings in which there are lots of pheromones, so there's lots of other animals around, even in the presence of food, they will reduce the rate and extent of their fat mobilization because there's kind of not enough food for the number of worms in that particular environment. And then the opposite is true. When there's low ASCAR3 levels, then fat metabolism is higher. And so we thought that was a completely unexpected and very interesting feature of regulation of fat by the nervous system. So around this point, we started thinking, all right, the gut is not innervated. We can ablate individual neurons. We can remove signaling. We can tweak neural circuits. We can activate and inactivate them. All of these pathways in data that I don't have time to show you are clearly tweaking ATGL combinatorially. But as I mentioned, the gut's not directly innervated, right? So how does this work? We had some preliminary evidence that neuropeptide secretion through dense core vesicles was going to be involved in this process. And so we decided to go ahead and conduct a screen. I don't know what I did there. Sorry about that. to identify these putative neuronal regulators. Okay, so there's about 120 neuropeptide genes in C. elegans, and we decided to screen them again in the context of serotonin because the combination of mutant screening, genetics and pharmacology was going to be really powerful. And so here's an example of our screen, right? So these are individual worms, but we don't, I'm just showing you individual worms, but we do this in a population basis, right? So we look at hundreds of worms. So this is wild type administered serotonin. We see the predicted and very stereotyped decrease in body fat stores, and each of these is an individual neuropeptide mutant. And we're basically simply asking, is there a mutant in which serotonin is now not able to trigger body fat loss, because that would become a candidate. We got a few sort of weak partial hits, and we completed the screen and held out for a robust hit, and it came 1. So these mutants do not show much fat loss upon exogenous serotonin treatment, suggesting that this gene is essential for serotonin-induced body fat loss. The name of this gene is FLIP7, and the name, again, in the C. elegans context is significant, but outside of that, the interesting thing about this gene is that it is an ancestral ortholog of the tachykinin family of peptides that were originally identified in the 1950s. FLIP7 mutants profoundly suppress serotonin-mediated fat loss, and that's quantified here, right? FLIP7 is expressed in a few pairs of neurons in the head of the animal, and we were able to isolate the necessity and sufficiency of FLIP7 in a single pair of neurons called ASI neurons. So this shows the rescue, and this shows the inhibition, indicating that it's sufficient. Expression from ASI neurons is sufficient to block serotonin-mediated fat loss. So, as I said, the gut is not innervated, right? So if you have a neuropeptide that is responsible for these effects, it could be working directly or indirectly. And so we were very keen to find direct regulators. And we thought that a direct regulator should be a secreted peptide, because we thought that it should be acting in the gut, right? So it's secreted by neurons and it acts in the gut. And so we developed an assay to ask whether this peptide, FLIP7, was in fact secreted. And the assay works as follows, right? There are cells within the C. elegans circulatory system called celomocytes that non-specifically endocytose anything that's in the body fluid. And by countermarking the celomocytes with GFP, we can measure and ask whether a secreted peptide is actually localized there, because it's a surrogate for what might be available in the circulation. And these are just controls to show that our method works, but it's published, so you can go read about it if you wish. Shown here is a wild type animal whose celomocyte is countermarked with GFP, and FLIP7 secreted from ASI has been tagged with mCherry. And what you can see here, I hope, is that FLIP7 is expressed FLIP7 is secreted and taken up by the celomocytes. Addition of exogenous serotonin greatly increases the secretion of FLIP7. TPH mutants that don't make any serotonin have very low levels, shown here, which can be supplemented a little bit with serotonin, but not as potently as in the wild type. Mod5 mutants, you'll remember, don't have the serotonin reuptake transporter, so they have more synaptic serotonin, and accordingly we see a constitutive increase in the secretion of FLIP7, as seen in the celomocytes. So through a series of studies that I won't be showing, we figured out that that FLIP7 is secreted in fluctuation and in proportion to serotonergic neural circuit activity. But what about the receptor, right? Here we got a little bit lucky. 2 receptors had already been described for FLIP7, and one of them, NPR22, turned out to be the key receptor that's essential for serotonin-mediated fat loss. FLIP7 and NPR22 double mutants retain fat just the way fat in the intestine, just the way each single mutant does. In this double mutant context, when we put FLIP7 back in neurons, but NPR22 is still missing, we don't rescue the phenotype. If we put NPR22 back in the intestine, but FLIP7 is still missing, we don't rescue the phenotype. It's only when in the double mutant context, the ligand FLIP7 is secreted from ASI and its cognate receptor NPR22 is present in the intestine, do we see a full restoration of serotonin-mediated fat loss. So FLIP7 and NPR22 function as the key ligand receptor pair that connects neuronal fluctuations to changes in intestinal fat stores. So that defines, the tachykinin system then defines the core neuroendocrine axis for neuronally regulated fat loss in the gut. Now, and I'll just show you what happens with overexpression of FLIP7. FLIP7 wild types that have an overexpression of FLIP7 have a constitutive decrease in body fat stores that's dependent on the presence of this enzyme ATGL. And this occurs not because of malaise or anything like that. It occurs because there is an increase in energy expenditure in the whole body of the animal, but we think that the phenotype is coming from the intestine because loss of the key enzyme that converts fats to energy blocks this increase in constitutive energy expenditure. So that was sort of nice and satisfying. But around this time, we started thinking about sort of the so what problem, right? So if intestinal fat stores are altered, so what is the consequence to the animal if it is high in intestinal fat or low in intestinal fat? In what way does it actually matter? And that naturally had us starting to think about, does this actually affect longevity? And so this also gave us an opportunity to address a conundrum in the field, which is that on the one hand, in disease models, not just in C. elegans, but across all species, in disease models, when there's ectopic deposition of lipids in peripheral organs, right? So for example, liver, too much adipose tissue, fat deposition in muscle, in the intestine, and so on, have been associated with reduced mortality. Right. And so in that context, if you convert those stored fats to energy by essentially just expending the energy, it's beneficial to longevity. Right. On the other hand, there's a whole host of studies showing that caloric restriction and decreasing metabolic rate is beneficial to longevity. Right. So on the one hand, we have data showing that increasing metabolic rate is beneficial, and then we also have data showing the decreasing metabolic rate is beneficial. And as we started thinking about this problem, we realized that these studies are done in totally different contexts. In one case, it's done in a disease context, and the other, it's done in a normal context. But in fact, no one had been looking at what happens in otherwise healthy animals if you dramatically increase metabolic rate and reduce body fat stores. What happens? Is that beneficial or is that detrimental to lifespan? And because we had this sort of genetic tool where the animal is otherwise completely normal, it's overexpressing FLIP7 and it has reduced fat stores because it has increased energy expenditure. Otherwise, it's a completely sort of healthy animal. So in this context, is there a consequence to longevity? And we thought that asking this question would sort of, again, fill that gap in our understanding of the relationship between lipid metabolism and longevity. And in data that I won't get into, but was recently published, my postdoc Nicole spearheaded a study in which we found that there is a counterbalancing mechanism that exists in the intestine to balance through a feedback loop the relationship between fat loss and longevity. And just to sort of briefly give you an overview of how that works, it turns out that overexpression or sort of continuous expression of FLIP7 stimulates fat loss by inducing ATGL, but it does so by repressing a highly conserved transcription factor called HLH11. And it's the worm ortholog of the mammalian AP4 transcription factor. And it turns out that HLH11 constitutively represses ATGL, but activation from the nervous system, either by serotonin or by FLIP7 via its cognate receptor, NPR22, relieves this repression and stimulates fat oxidation and mitochondrial respiration. However, simultaneously, it invokes a mitochondrial stress response through the canonical and well-studied transcription factor ATFS1 that was originally discovered by the Haynes group. And it turns out that ATFS1 is a sensor of mild mitochondrial stress and communicates back to the nucleus from the mitochondrion to sort of, at least in our context, titrate the extent of fat loss that is that the mitochondria are able to handle. So in other words, there's a sort of stress induced by fat metabolism and mitochondrial respiration within the mitochondria, and that stress is sensed and regulated via the ATFS1 HLH11 interaction, so that it's titrated in such a way that it doesn't affect longevity. And if you take this loop away, there's now a cascading shortening of lifespan that is depicted here, right? So on their own, this pathway is completely latent, but if you stimulate fat loss, but take away the counterbalancing mechanism, that is, you take this arm of the pathway away, and you see a dramatic increase in death. Very early in life, where very little death is seen in both wild type as well as other mutant conditions. And so this told us that fat metabolism in otherwise a robust fat metabolism in otherwise healthy animals is completely fine. That is to say, it does not shorten lifespan as long as the mitochondrial stress response and this communication between the mitochondria and the nucleus and this transcription factor back to the adipose, the lipid droplets, as long as this communication is in place, there's no change in lifespan. But if you shorten this arm of the pathway, you inhibit fat loss. If you remove this arm of the pathway, you reduce lifespan. And so we thought that was sort of an interesting sort of with interorganeller conversation, if you will, that's required in the intestine to regulate lifespan. So while these studies were going on, I started thinking that how does the nervous system, so yes, it's responding, these neurons are responding to the environment and regulating fat stores, but how does the nervous system know how much fat there is in the first place, right? There has to be some homeostatic set of mechanisms that inform the nervous system about what's happening metabolically, right? And we had some sort of evidence that kept building up and kind of wouldn't go away, right? So one of the first things we had observed when we had done our oxygen sensing studies, and this was inspired by the anatomy of these oxygen sensing neurons themselves, right? Even though they sense environmental oxygen, their dendrites are actually within, and their cell bodies are within the internal milieu, they're within the circulatory system. So that always suggested to me that they're sensing something from within the animal. There is some sense of perhaps interoception, sensing of the internal state. And so we were always very curious about that. And we did kind of a super simple experiment, right? We simply inactivated a host of enzymes. I'm only showing one as an example here, but these are enzymes that are part of the fat synthesis pathway. The one that I'm showing you here is called acyl-CoA carboxylase, which is rate limiting for taking acetyl-CoA and creating malonyl-CoA. That is the foundational metabolite for building fat stores. And so when we inactivate acyl-CoA carboxylase just in the intestine, right, you can see a pretty dramatic decrease in body fat stores because we're not allowing the animal to make fat. And one of the things that we noticed was, video's not working. I'm sorry, but if this video was working, you would see that when you activate these neurons with oxygen, you see sort of a bright response. But if you remove the fat stores or you deplete the fat stores from the intestine, we see a profound decrease in the extent to which these oxygen-sensing neurons are able to be activated. And so we'd always thought that was really fascinating, that if you simply deplete the fat stores, these neurons are somehow responding. And we did a variety of controls to make sure it wasn't an artifact of something. We tested many different fat regulatory enzymes and sort of kept getting the same result. Another thing that we did was to simply look at our secreted neuropeptide, right? We looked at FLIP7 and asked the question, if you deplete fat stores, what happens? And again, we did it using both acyl-CoA carboxylase and SREBP, which is a canonical transcription factor that's responsible for activating a cascade of enzymes and pathways involved in lipid synthesis. And in both cases, we saw something completely surprising and paradoxical, which is if you inactivate these enzymes, thereby depleting the gut of its fat stores, it somehow increases FLIP7 secretion, suggesting that there's some communication from the intestine back to the ASI neurons that are responsible for secreting FLIP7. We saw this not just at the level of sort of neuronal activity via GCAMP or neuronal output via FLIP7 secretion. We even see it at the level of overall behavior, that if you fat deplete animals, they act as if they don't have enough food, right? So this is an assay, it's called the roaming dwelling assay. In this assay, if you take a well-fed, fat-replete animal and put it on a lawn of food and ask how much does the animal forage for food, right? What you will see is, for the most part, the animal is kind of dwelling, right? It's hanging out where the food is, depicted here, right? This gray patch you have to imagine is the patch of bacterial food. The animal is, for the most part, 70-80% of the time it's dwelling. It's not roaming very much. It knows there's food available. But if you fat deplete the animal, it roams twice as much. And roaming is an indication of, I'm looking for food. There's not enough food here, right? It's an indication of searching for food. And if you deplete the fat stores from the intestine, the animals roam twice as much as a fat replete. This is an otherwise completely wild type, right? So there's clearly some internal sensation of food or fat or nutrients that is missing, that is causing this behavioral shift. So all of these sort of observations got us thinking about the gut as both a sensory organ, as well as an organ that is relaying some internal state or some sensory information that the nervous system is able to see. And so we decided, sort of in terms of, how do we start? We decided to take a translatomics approach of the intestine, right? So we can, again, using cell type specific markers, get into different parts of the intestine and simply ask the question, in the fed state or the fasted state or the refed state, what are the genes or what are the transcripts that are specifically translated into protein, right? So we're not interested, of course, we would be unsurprised if hundreds and hundreds of genes changed if you fast an animal or refeed an animal, right? That would be unsurprising, but we are particularly interested in those transcripts that are acutely required for the transition from the fed to the fasted state and back. And so from our initial, this is all sort of like hot off the press data, we're still trying to figure out how to analyze the huge scale and scope of the information that we're getting from this. But our very early data suggests that even a brief fast where the intestine is depleted of fat stores, then of course we're looking specifically at transcripts in the intestine. We see, unsurprisingly perhaps, that oxidation reduction processes are involved, defense responses are involved. There's of course fatty acid metabolism. We thought it was particularly fascinating that determination of adult lifespan genes are coming out from some of our GO analysis. This phosphorylation group is very indicative of potential signaling roles, which we're interested in exploring. And so innate immune response genes are involved. And so we're using this approach to try to get at what are the particular sets of signals and how might they be working for the gut to relay information back to the nervous system. What is the nature of that information? What does it look like? In what contexts is it salient? And so through some of our early screens, we have already begun to identify some pretty tantalizing peptides. C. elegans has many insulins, some of which we think are responsible for gut-to-brain communication. We also think that other gut hormones like the C. elegans ortholog of cholecystokinin and perhaps others may be playing very interesting and informative roles in sending information from the gut back to the nervous system. And so our current hypothesis is not so much that the brain regulates metabolism or that the metabolism regulates the brain, but it's really the communication and the conversation between these different organs itself that is foundational for driving both behavior and physiology. So in the coming years, we're very excited to decipher some of these pathways and the way in which they may be modulating. Just a brief sort of last bit of data to say that we are just starting to look at the ways in which these pathways might be conserved in mammalian models, and to our absolute delight, this is a cross section of of the intestine in the mouse, and it's from an explant, and we see that addition of serotonin or its inhibition of ATGL, the enzyme that converts stored triglycerides to free fatty acids, does seem to regulate intestinal lipid metabolism. And so we have a model in place that we are currently testing. So I'll just end with sort of where I started, right? Where do we stand in terms of our thinking about the relationship between genes and the environment in regulating body fat homeostasis? And so we think that serotonin and the tachykinin pathway will play very important roles. We're sort of starting to make some inroads in our understanding of how they may be regulated and where it may be useful to intervene. FGF21, done through the work of others, is also emerging as a potentially interesting candidate. And so we think that this sort of range is a combination of food and bacteria, gut hormones, and genotype. And so with that, I'll stop, and I would like to acknowledge many people, both current and past, my many former postdocs and students and associates who did really amazing and beautiful work, without which I would not be standing here. Our lab studies neurons and fat. As you can see, members, my collaborators, and the broad and robust and beautiful scientific community at Scripps. With that, I will stop, and I'll be happy to take any questions.
Thank you so much. It was a really, really lovely talk, and I really liked your conclusion. I mean, I also believe the same, that it's not about which organ commands which, it's about the hub and a communication hub, and in the end, that it's the output that we're trying to measure, which is kind of difficult. Mm-hmm. Really, thank you so much. Oh. Okay, so we have a couple of questions. So the first one from Zita Santos. She says, I was wondering if the changes in oxygen sensed by these neurons could be also derived from differences in beta oxidation of the fatty acids and not just from external differences. And if they could sense both internal O2 release use as well as external.
So did I hear correctly that she said BAT? Like brown adipose tissue oxidation?
Beta oxidation.
Yeah, so right. So this is actually like an interesting question, right? So it's clear to me from the genetics that the sensing of environmental oxygen is a big part of what's going on in terms of this relationship, both behaviorally and metabolically. But the catch is this, right? These neurons have cell bodies, and some of them have their dendrites also that are projecting into the circulatory system, right? And the worm itself is probably able to, there's probably oxygen in the internal milieu within the circulatory system that is much higher than what it would be in mammals, for example, right? So that they're sensing oxygen is clear. but to what extent they're sensing pure environmental oxygen versus the conversion of that environmental oxygen to what's in the circulation isn't known, right? I would speculate that it's probably sensing internal environmental oxygen, but that internal environmental oxygen is reflecting what's on the outside, if that makes sense.
Yes, I see. Thank you so much. We have another question from Hosanna. And the question is, could you please talk about the connection between mental diseases from anxiety, depression, etc., all the way to schizophrenia being connected to gut health? This is a bit more broad, but
That's a very broad question. And actually, you kind of hit on a very important nugget, haven't you? Because there's been a lot of interest in this. And I'll speak from within the context of serotonin, which is the context that I think the most is known about and that I understand the best. That gut serotonin seems to play really important roles in anxiety, in emesis, for example. And so there's some potential applications in food, eating disorders, mood disorders. The extent to which, so if you look at absolute levels of serotonin, right, there's 100 times as much serotonin made in the gut as there is in the central nervous system. And yet it's clear that there's some interplay between those two at the phenotypic level, right? And so it's a completely fascinating question, that this idea that we think that these mood disorders are in our head may actually be mood disorders that originate in lack of communication between the gut and the brain. But beyond speculating about these potential roles, I think we now have genetic tools to get into mechanistic aspects of this, right? So for example, with the single cell sequencing, both of the intestinal epithelium as well as, I guess it's not single cell sequencing, but at least of sequencing, for example, of the cell bodies of the nodose ganglia that are the cell bodies of the vagus nerve that innervate the upper GI tract. We already know, for example, that the 5-HT3 receptor is sitting in a subpopulation of the vagal ganglia. What are they doing there? Well, gut serotonin is one of the sort of major modulators, we think, not only of GI metabolism, bone function, but may also have a role in anxiety, may have a role in feeding, may have a role in mood-related behaviors, right? So we have now the tools to start to get into that. So I think the next few years of work across all model systems in studying this, studying the gut from the point of view of behavior, studying the brain from the point of view of metabolism will be very, very fruitful.
Thank you so much. Really cool. I actually have a question of myself. I think we still have maybe a couple of minutes. I was wondering when you were looking into longevity and the effects, I found interesting that only when these manipulations were done in early days that you actually saw an effect. Later on, it actually seemed the opposite. I'm not sure if I missed something or if there is a reason for this.
Let me see. Let me pull that data up. Here we go. If I'm not mistaken, you were talking about this slide, yes?
Exactly.
So some of this is the opposite effect is actually because more animals have died earlier on, right? So this is because most of them are dead at this point, right? We try to, work with about 100 animals, and so if you see this, it's because they're nearly all gone already, right? So the big phenotype is that very early on, when there's such a dramatic fat loss, if you remove the compensating mechanisms, you see this kind of more than double increase in deaths, right? So there's a 50% shortening of lifespan, but the reason you see this here is because they're already dead.
I see. Thank you. Thank you so much. Okay, I think with this we're done. We will now move to the Q&A session in Zoom with Dr. Supriya. And I would like to thank you all for being with us today. Next week we will have a break, but the following week we will hear from Dr. Christoph Scheierman telling us about sympathetic control of lymph node function. So with this, I wish everyone a great week, and thank you once again, Supriya, for your time. It was a really, really amazing
Oh, fantastic. Thank you so much. It was my pleasure.
talk. Bye, everyone.